How gene editing could stop malaria. The mosquito revolution. A single genetic change in mosquitoes could revolutionize malaria prevention. Imagine a tiny tweak in a mosquito's DNA that could stop one of the deadliest diseases on Earth, malaria. This isn't science fiction. It's a breakthrough recently discovered by researchers who found a way to disrupt the parasite's life cycle within the insect. But here's where it gets controversial. While the idea is groundbreaking, its real-world application raises serious questions about safety, ethics, and unintended consequences. Malaria claims over 260 million lives annually, yet the parasite's journey to infect a human is anything but simple. It begins when a mosquito bites someone infected with Plasmodium parasites, bringing the pathogen into its gut. The parasite then embarks on a perilous voyage through the mosquito's body, navigating a maze of tissues until it reaches the salivary glands, the final destination for injection into a new host. Now, a team of scientists has found a way to sabotage this critical step. Using gene editing tools like CRISPR, researchers altered a single amino acid in a gene called FREP1, which plays a dual role in both mosquito feeding and parasite survival. The result? Mosquitoes with this genetic modification have 5 times fewer parasites in their salivary glands, with many showing none at all. The insects themselves remained healthy, suggesting the change didn't harm the mosquito. Changing just one amino acid and preventing parasite transmission is a game changer, says entomologist Fred Gould. It's a reminder that small changes can have massive impacts. The next step, scaling this discovery to real-world populations. Scientists hope to use a technology called a gene drive, a genetic tool that overrides the usual 50-50 inheritance rules to spread the modified gene across entire mosquito populations. In lab experiments, the beneficial change spread to over 90% of the population in just 10 generations. If successful, this could transform malaria-carrying mosquitoes into harmless ones, potentially wiping out the disease in regions where it thrives. But this isn't without risks. Critics warn that gene drive could have unpredictable effects on ecosystems. These tools could alter genetic landscapes in ways we don't fully understand, says Dana Pearl of Friends of the Earth. Once released, the changes might persist for decades, disrupting natural balances. Some fear that the genetic modification could inadvertently affect other parts of the mosquito genome, leading to unforeseen mutations. Others worry that eliminating mosquitoes entirely could harm ecosystems, as these insects serve as food for birds, bats, and other species. To address these concerns, researchers are exploring alternative strategies. One approach targets mosquito fertility, aiming to reduce populations without harming ecosystems. Another focuses on engineering mosquitoes to produce proteins that directly attack the malaria parasite. However, the FREP1 method stands out because it doesn't introduce new genes. It uses a naturally occurring variation that already exists in some mosquitoes. This is a more acceptable option for communities wary of genetic engineering, says molecular biologist Anthony James. It's like using a naturally occurring bug fix instead of a synthetic one. Still, the road to implementation is long. Field tests will take years, and local governments and communities must approve any release. We need this tool, says biologist George Demopoulos. Traditional methods like bed nets and antimalarial drugs aren't enough to end the disease. We're facing a public health crisis that demands new solutions. So what does this mean for the future of malaria control? While the genetic tweak is a major step forward, it's just one piece of a larger puzzle. As scientists refine this technology and address its risks, the question remains, can we balance innovation with caution? And if we do, will this be the key to ending malaria, or just another step in a longer, more complex journey?